Inflammex is an institute for inflammatory diseases, is a network of excellence, is built to understand inflammation and fibrosis. What's lacking in the country is something like this network that bringing together different units, different researchers to get together and to create together new ideas and new projects to understand, but also to generate new treatments for inflammation. The idea that we had is to develop comparative cohorts, including prospectively 50 patients for each of the disease, so going from nephritis, hepatitis, inflammatory bowel disease, diabetes, arthritis, asthma, fibrosis, and vasculitis, and to create those large comparative cohorts to address common markers and specific markers and study their involvement in the pathology. One of the goals of Inflammex is to generate this kind of models to mimic the inflammatory diseases and to use them as proof of concept for new biomarkers. And we have an example on that. Indeed, studying IgA nephropathy patients, we identified abnormalities in two proteins, the immunoglobulin A and also the receptor of the immunoglobulin A. And indeed, we create animal models expressing the human IgA and the human receptor. And these animals develop the disease. Not only they develop the disease, they also express markers that are being able to identify that are common to other diseases, the intestinal disease such as celiac disease. We published this observation. Immediately an American company contacted us because they developed a product that only works on the human protein. And indeed it works. We have extremely interesting results, really clearing the disease. So it's extremely encouraging for putting this drug in a phase 1, even to phase 2. We developed a new antibody using a concept that was kind of regulating inflammation and anti-inflammatory reactions. And when we generate this kind of antibodies in the mouse, we're able to prevent development of nephritis, of asthma, of arthritis. And a startup, a spin-off of one of the teams of Inflammex, is now developing this product and is also collaborating with partners for patient cells with asthma to try to see whether this antibody is also active in patient cells before moving to phase 1.